In this video, we'll be dealing about the perfect concepts of glycolysis, right? So let me read what's present in our notes. It's written that it occurs in all cells. Okay, fine. At the time, all the time with oxygen and mitochondria. Okay. Occurs in cytosol. Okay. Main aim production of ATP. Right. So now let us understand the glycolysis concepts, right? So right now it's already time. Right now, suppose if you eat rice, right, in your dinner, so now let us understand that the rice you are consumed or you are going to consume, have this leads to a production of ATP. Rice is nothing but the glucose, right? So now what happens here is, the moment it breaks down, right, and there's a formation of glucose right now in your blood. Now this glucose enters the cell. Right now this is glucose. Now let us understand that there is something going on with the glucose. We have to break that glucose, right? We have to break the glucose. So now once we start breaking this glucose, right, we are doing it for the ATP production. So now let us understand that there's a glucose at this aspect, and now let us see the final equation first. Glucose plus 2 NAD plus 2 ADP plus 2 PI gives 2 pyruvate plus 2 ATP plus 2 NADP plus 2 H2O plus 2 H. So what is it trying to tell us? So what is the 2 use ATP and 4 ATP, right? So now let us understand the glycolysis cycle perfectly, right? So let me take this section. You already know the mnemonics now. You already know that girlfriend dog bit my pant, or non-veg guys. Okay, fine. Let us understand that. So let us take the first word of G from our mnemonic. So let us take the first word G. So from the girl, I have told you girl, G for G square. So let us take the first word G and write it here. Right. So this is the structure of glucose. When I see the structure of the glucose, now let's see what's gonna happen with the glucose. Right. Let us see what's gonna happen with the glucose. We have to break that glucose properly. Now for the conversion, we know the first part of the mnemonic, that is G square, right? One is glucose and other is glucose 6-phosphate. Now let us understand that. Now if you observe the diagram in this aspect, right? If you observe the diagram here, this is carbon 1, carbon 2, carbon 3, carbon 4 and carbon 5. Now if you understand the concepts here, now if you compare it here, compare with this diagram, so what is the difference here? The difference is, what is the difference? The difference is at 6th position. If you observe at 6th position, there is something which is extra in this compound, right? That is glucose at 6th carbon, 6-phosphate. Now before there was no phosphate, and now there is an addition of phosphate, right? Before you don't have a phosphate, and now there is an addition of phosphate. Now how the what is the reason behind? Yes, you got some enzymes, right? That catalyzes the transfer of phosphate group from ATP to specific molecule. Now you got an enzyme known as hexokinase. What is hexo? Hexo is 6 carbon molecule, right? Hexose. Now what is kinase? Kinase is an enzyme that catalyzes the transfer of phosphate group. From ATP to specified molecule, and the molecule which are specified is glucose. So now we are grabbing this enzyme hexokinase is grabbing the phosphate from the ATP. from the ATP. Once it grabs the phosphate from the ATP, and now you can see the glucose initially with no phosphate. Now the addition of phosphate has taken place because the ATP has donated phosphate to the glucose and it has become ADP, and now you have something known as glucose 6-phosphate. Glucose 6-phosphate. And this is what you have to understand regarding the first step. The easy way of understanding the concepts is know the mechanism of these enzymes. The mechanism of the enzyme hexokinase is, what is that? It's a transfer of phosphate group from ATP to a specified molecule here. Right, and now we know the concepts of the first mnemonic, that is girlfriend, girl, right, G square. Now let us see friend is F square. So now let us understand that, right, this glucose 6-phosphate is converted to fructose 6-phosphate. It is converted to fructose 6-phosphate. Now the name itself is telling the glucose 6-phosphate, right, just it has converted to fructose 6-phosphate. Nothing, there is nothing addition, there is no phosphate, there is nothing addition. You have just a conversion of glucose has become fructose. And such type of mechanism which is coordinated by an enzyme known as isomerase. It is coordinated by an enzyme isomerase. What is isomerase? Isomerase is an enzyme which catalyzes the conversion of specific compound. Yes, it's catalyzing the conversion of this specific compound to isomers. Now glucose 6-phosphate, it is converted to isomer of fructose 6-phosphate. So for the conversion of one isomer to other isomer, you need an enzyme known as isomerase. You need an enzyme known as isomerase. So that is what you have to understand first. You have to know the concepts. Let's know the concepts that makes your glycolysis or any cycle, right, easier. And next let's understand, let's take the next word from the F, that is fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate. Now let's not try to see the enzyme here. Let's see only the conversion here. In this conversion, if you see fructose 6-phosphate is converted to fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate. Now, what is the change between the fructose 6-phosphate and fructose 1,6-bisphosphate? In fructose 1,6-bisphosphate, there is extra phosphate added, extra phosphate. That is the reason the fructose 6 has become now fructose 1,6-bisphosphate. If you see the compound here, this is a 6 carbon. You can see the phosphate before, and after the addition, right? After this addition of phosphate group, not only on the 6th position, the phosphate is present on the first carbon also, and this is mediated by an enzyme, right? Same enzyme what it was mediated before, right? That is also kinase. This is also kinase, but the reactant here is what is that? Fructose 6-phosphate, right? Yes, phosphofructose. You can write it, right? Phosphofructose kinase. Now who is donating? It is the ATP which is donating phosphate. Kinase is nothing but taking a phosphate giving to the compound, right? And ATP is donated to ADP. ATP is donated to ATP. And now let us understand, we knew the concepts of right now we know girl, friend. Now let us go with the dog, D-O-G, right? Let's know the concepts of dog. What is D? Right, if you observe carefully, right, if you observe carefully here, the fructose 1,6-bisphosphate, which is a 6 carbon, molecule, now it has divided into 3 carbon molecule, 3 carbon, and this is, right, this is 3 carbon, and now it has divided into 3 carbon into 2 molecules, right? And now who is that enzyme? Let's rule out first. Now enzyme, you think that might be kinase which divides? No, the function of the kinase used to donate the phosphate. Do you think it is isomerase? No, isomerase is used in isomerization, right? Now what do you think? Yes, you got an other enzyme known as aldose. We have got an other enzyme known as aldolase, right? And this aldolase enzyme, right, this is aldolase. And this enzyme aldolase, what is the function of this enzyme aldolase? The function of this enzyme aldolase, it breaks the sugar. Yes, it's breaking the sugar now into 6 carbon into 2 carbon, right? Sorry, 3 carbon into 2 molecules. You got a 3 carbon and a 3 carbon, Right, and you have to remember that since dihydroxyacetone phosphate, the compound here, if you see the functional group, it is keto group, and if you see glyceraldehyde 3-phosphate, you can observe here it is an aldehyde group. That means they both are isomeration. So for the conversion of isomerase, what is the enzyme? Yes, isomerase. Yes, isomerase. What is the reactant? Yes, this is an triose phosphate. It's 3, right? 1, 2, 3 triose, and we have got a phosphate here, triose phosphate, triose phosphate. And this is what you have to understand now. Now we finished with the first concepts of D, and we also finished the second concepts of G, right? Now let us understand that how this G glyceraldehyde 3-phosphate, right, now this glyceraldehyde 3-phosphate, what's gonna do? That means we know the concept of girl, friend, dog. Now let's go with the concepts of bit, BTT. Now if you observe carefully, right, if you observe carefully here, you got something, right, glyceraldehyde 3-phosphate. Now we have got the mnemonic, I have told you, bit, BTT. So if you see the mnemonic of bit, BTT, what is B? B is bisphosphoglycerate. That is 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate. And did you observe something here? Here we had only one phosphate in glyceraldehyde 3-phosphate, and now we have 2 phosphates. That means something has happened. Some addition, some subtraction had happened. And what is that? Yes, there is a phosphate. There might be some addition of phosphate. Yes, you are right. There is an addition of phosphate, right? There is an addition of phosphate. And due to the addition of phosphate in this step, we have the extra phosphate here. Now, let us understand that to add the phosphate, right, to add the phosphate here, we have to remove the hydrogen atoms. For removing the hydrogen atoms, right, now let us understand that there is an enzyme which removes the hydrogen atoms, and those are, they are removing dehydrogenase. The enzyme that catalyzes the removal of hydrogen molecule is dehydrogenase. It is removing hydrogen atoms from the glyceraldehyde 3-phosphate, so we call it as glyceraldehyde phosphate, right? Glycerophosphate dehydrogenase, remember, right? Now fine, it's removing hydrogen from glyceraldehyde 3-phosphate. Now who is taking that hydrogen? And it is NAD which is taking the hydrogen atoms, and it is forming NADH, right? Now let us understand, we knew the concept of 1,3-bisphosphoglycerate. Now in the bit BTT, now let us understand the second concept of T, that is 3-phosphoglycerate. Right, if you observe here, we had 1,3-bisphospho, means we have got 2 phosphates, right? Now there is only one phosphate. So something had happened. That means there is one phosphate which is removed, right? Which phosphate is removed? You can observe the structure here. You have got a 1, 2, 3 at one position, and the third position you got a phosphate, and see here, there is no phosphate at first carbon. Phosphate at first carbon, and that is removed by ADP is converted into ATP. I told you, right, any change with the ATP, right, the enzyme you're gonna use here is kinase, right? The enzyme we are gonna use is kinase. We have to understand the concepts first, and this kinase, I have already told you, it plays a vital role in the phosphate, right, in the phosphate reactions. So understand that ADP has converted into ATP. So now we finished in the mnemonic of girlfriend, dog, bit, BTT, and we have got a last T for 2-phosphoglycerate. Now are you observing something here? Are you observe carefully that you have something known as 3-phosphoglycerate and 2-phosphoglycerate. There is nothing addition, there is nothing is going to happen, but the number has changed, from the third to second. That means do you think enzyme kinase is no, kinase function is ATP. Do you think isomerase? No, that is function for isomerase. Yes, you got a special enzyme known as mutase. This mutase plays a vital role in shifting in shifting the position of the phosphate from the third carbon to the second carbon. So we have included the enzyme mutase. We included the enzyme mutase. And now we knew the concepts of girlfriend, dog, bit. Now let us know the last mnemonic of for the non-veg, that is penis, and for the veg it is pan, right? And last here is phosphoenol pyruvate. Phosphoenol pyruvate, that is 2-phosphoglycerate is converted into phosphoenol pyruvate. So if we observe carefully, the enzyme used here is enolase. So you can observe the end product enolase. Yes. And now we have come to the last point of this cycle, that is phosphoenol pyruvate is converted to pyruvate. It is converted to pyruvate. Now this is the basic. You have to understand the concept of the mnemonic of girlfriend dog bit my penis for the non-veg, girlfriend dog bit my pant for the non-veg. So now we have come to a basic structure. We knew we are fucking concept. Right now we knew the concept. Yes, we know what is G, we know what is glucose 6-phosphate, yes, we know what is fructose 6-phosphate, we know what is fructose 1,6-bisphosphate, and we know dihydroxyacetone phosphate, we know glyceraldehyde 3-phosphate, yes, we know 1,3-bisphosphoglycerate, 3-phosphoglycerate, 2-phosphoglycerate, phosphoenol pyruvate, and pyruvate. And this is what you need. This is how you have to study for understanding each and every word, right? And now let us understand the energetics, in the sense the ATP is produced in this cycle. Now just a glance now. That means we are not going to by heart anything. Now just observe. Let's see where the word kinase had come in our cycle. Right, the kinase had come with the first cycle. So can I tell one ATP is used here? That means there is some ATP function either right now, but based on the kinase we don't know that ATP utilized or it's given. But if you observe here, one ATP is utilized in this step. This is -1. Why? Because this glucose is pulling out phosphate from this ATP. It's pulling out. So that is why you have got -1. Now let us understand. Let's see the same reaction where it is going on. Yes, the same reaction if you observe this kinase. Yes, there is utilization of one phosphate. So that means there is one negative ATP because it is using. Right, so that is the reason I have wrote -1. And now, are you finding anything in the aldolose? No. Are you finding anything in isomerase? No, our function is not that. Yes. Now there is an enzyme known as dehydrogenase, right? Dehydrogenase. Yes, you have to think something. We think something. Yes, hydrogenase. Yes. Now I got the concept that dehydrogenase, it is removing hydrogen from this compound and it's adding, right? It's adding hydrogen to NAD, and now there is a production of NADH. And these NADH go to the electron transport chain. And it tells tata, bye bye. It goes to electron transport chain, and now you have to observe for one NADH there is production of, right, 2.5 ATP. Yes. Now let us see the next kinase. Yes, we got kinase. That means now ATP is produced. So can I tell it is +1? Yes. Now mutase, no, I can't touch that because there is no kinase, there is no dehydrogenase. No. Can I speak with enolase? No, I can't speak with enolase. Why? Because the ATP is not produced here. So can I speak with phosphoenol pyruvate? Yes, you have to speak because you got something known as kinase. And this kinase, so addition +1. Right. So now we got a basic concept that there is 1, 2, you got 2 ATP, right? There is a utilization of 2 ATP and production of how many, right? 2.5 plus 1, because in this equation and this equation plus 1. So how much is that? It is 4.5. But now let us understand. Now let us understand the concepts here. What concept you have to understand here? Now I spoke only about this one glyceraldehyde 3-phosphate. Now you have to understand that even you have other molecule known as dihydroxyacetone phosphate, even that also converts, right? Even that also can go into this cycle. If this cycle has gone, then can I tell the ATP produced in this sequence is also same? So can I tell 4.5 for one cycle, then other 4.5 for another cycle? Another cycle in the same? This also goes. Right, this also converts into glycerol here, so you get it here, right? So now there is total production of 9 ATP, but you have got 2 ATPs are utilized here, 1, 2. So what is the answer now? 9 minus 2 is 7 ATP. So that's what it's written in our energy sequence. If you see energy sequence, that is what it's written. At the first equation 1 ATP is used, say third equation 1 ATP used, and sixth equation, that is there is formation of 2.5 into 2 NADH, right? NADH 2, so you got a 5 here, right? So now same thing, 1.2, 2. So total I have told you 9, 2 are utilized, and you got it of 7 ATP. This is what happens in aerobic cycle, because if oxygen is present, then it goes for electron transport chain. Now let us understand what happens in anaerobic. In anaerobic, it can't go to the electron transport chain. So let us cancel this NAD. Right, NADH, the electrons which are produced here. Now the only electrons which are produced are at the 7th step and the 10th step. So you got 2 electrons, so you have got 4 here, and 2 are utilized here. So 4 minus 2 is 2 ATP. 2 ATP are generated in anaerobic reaction and anaerobic reaction. So this is what you have to understand, that don't get confused with the anaerobic. In anaerobic there is production of NADPH, but there is no utilization of NADPH in if there is no oxygen. So that is the reason I didn't write this column. That is the reason I didn't speak about this NADH, right? I only spoke about what is produced here, right? So this is what you have to understand, and this is what our whole equation, right? This is what they tell us, right? This is what they tell us, and you can observe here minus 2, that means 2 ATPs are utilized, 4 are produced, 2 NADPH, they go to the electron transport chain, mitochondria, and there is a production of 2.5 or 1, that is a 5, so total of 9, and 2 are utilized, right? That means total 7. In anaerobic 4 are produced, now 2 are utilized, so net is 2. So that is all about this first concepts of glycolysis, and let us continue in the next session regarding the next what's written in our notes. Thank you guys, hope you understood.